shown over here. These strange structures no one had seen before. The monomer looks like this in the beginning, and then at very few time points, or very short times after initiation of the reaction, you see what we call fuzzy balls, these spherical aggregates, and then what we call the acuna. So what's the acuna? The acuna is shown here in more detail. It is this kind of structure which is formed of synuclein only, half a micron more or less in diameter, in which these fibers then are forming. Now these are not amyloid fibers. There's some other extended molecule or molecular structure, but you can see by analysis over here that they start on these empty shells, if you like, and then they form and extend and are liberated. And when they are liberated, they form what are called, these are another example of these things. They form these so-called fuzzy fibers. And they're called fuzzy because they have this external material, which we think is monomeric synuclein bound to these extended structures. The question is, are these toxic? Well, they look toxic. We don't know whether they're toxic yet. This is the next story to tell you at another time. So we made a catalog of all these things. This was just published in Biophysical Journal. And another interesting feature is that if you do the reaction, the initial reaction at low temperature, at high concentration, you get these acunas. And by the way, I said they're acunas because it's the construct of amyloid and cuna. Cuna, you know what that means. And we thought that it's time to do something about the imperialism of the English language in science. And so this is a Spanish word that we're contributing to this field. So these are latent acunas, and if you expose them to higher temperature, they start making fibers. So it's kind of a neat thing, and we're trying to study this in detail and get more information on it. We were able to do cryo-electron tomography with the lab in Baylor, and showing the inner structure of these acunas, and you can see fibrillar structures, which are probably aggregations of oligomeric forms of synuclein. This is very interesting and probably very important because it's the genesis of the first structures that lead to something of interest and perhaps toxicity. And then we have a big scheme. I don't have time now to go into this, which we divide into 3 stages: the molecular stage, the colloidal stage, and the fibrillar stage. And we think this is a new concept in the field, and that's why this is a new paradigm, because this is where the action takes place before you start forming these amyloid fibrils that are probably don't have much biochemical repertoire. So this you can read about in the Biophysical Journal, and this is a follow-up story with a super resolution showing that these structures may be found perhaps in cells. Well, now I don't have time. I guess I oversensed what I could do about 2 other things that I wanted to show you. That this has to do with membrane binding, using another probe of this ESA family to show the interactions of synuclein. And just let me indicate that in all of these kinds of studies, it's very easy to perform an experiment because these spectra change so well. So you can put the label anywhere you want to in the molecule, and positions are shown over here, and you can expose it to membranes and show what the influence is of size and shape and charge and curvature and so forth. This has been published in the JBC, and you can do kinetics and stop flow even, and get very nice information that is not available by any other technique that I know. And the most recent thing is to show that in the case of synuclein, you get binding very fast to a charged molecule, for example, a negatively charged vesicle, whereas the uncharged lipid vesicles bind more slowly. And I think what we have found in this study is that the initial binding is electrostatic. The molecule simply binds by that means and then rearranges to form structure. And this is new information. It's thought that this molecule becomes alpha helical, and we think we have now a kinetic view of that process, the first evidence for that in the literature, and that takes about half a second. It takes half a second to form the alpha helical structure after the protein bounds, probably as an unstructured molecule. You can also look at migration between different things, different kinds of vesicles, and you can determine that the orientation on the membrane depends upon the lipid to protein ratio, so that's very important probably in biological sense. And if you modify the protein, for example with SUMO, you get different effects depending on whether you're dealing with neutral membranes or negative membranes. So I'm almost finished, Pancho. All right, so there are conclusions that binding to negatively charged bilayers are driven by electrostatic forces. Neutral bilayers depend upon membrane defects. All right, then you can compare Alzheimer's disease and Parkinson's disease using a molecule that we label with this ECIP dyes, but as an amino acid replacement, this is very nice because you get a molecule that's very compact, and you can do the same kind of things with that. In this case, A-beta prefers rigid membranes. Synuclein prefers disordered membranes. And it's quite interesting to see the comparison on these kinds of different membrane type of domains, and we can show that by temperature adjustments, which is the preferable one for either synuclein or A-beta. So A-beta requires the L-beta phase of the molecule. Yeah, Pancho, just let me. So I think the take-home lesson here of interest is that when A-beta binds to membranes, it does not form an alpha helix. So this is against much of the reported literature, but we think it's based upon good evidence, and we see no formation of secondary structure when A-beta binds to a lipid bilayer. And this is important in the probably the biology of this disease. And there are comparisons I can make, but I won't go into that. I just want to show a couple. I'm sorry. These people are working here at the Laboratorio Max Planck at the university, postdoc Federico Fuentes and a graduate student Jimena Martinez and Fernando Guaymas, who's an undergraduate, and he's become now an expert in single molecule fluorescence, microscopy, and super resolution microscopy. I think it's a great thing that we can offer undergraduates as well as PhD students and postdocs a place to work. And they have looked now at the effect on mitochondria of synuclein binding. This is important because of course, the ROS involvement in these diseases. And we have shown that if you form dityrosine, I think Claudio Fernandez will speak a little bit more about this, you get things that you form species, cross-linked species that are inhibitory of aggregation, but also are toxic to cells. So there's an indication that an oxidation of alpha-synuclein leads to toxicity. This must be important in disease. And if you go to the mitochondria, you can ask whether the synuclein binds to that, and it does. And when it binds to that, it influences the function of the mitochondria. And how it binds depends on the concentration. If you go to high levels of the protein, you get inner binding, and it looks like the inner membrane is affected because you can do STED and super resolution on this. You can show that the activity of the cytochrome oxidase complex 4 is reduced. So this is a very interesting finding, and it's work in progress. The final thing, so I can't get to that, or else Dr. Barrantes will throw me off the podium. It has to do with the study of PD-derived induced pluripotent stem cells, and these are the people involved in Göttingen and also Federico Fuentes here. This is very important because it's being supported by the CERM Institute in California and the BMBF in Germany, and now this has been extended to Brazil and Argentina so that the Agencia here will also support a conjoint project with CRM. So we want to take advantage of this, and we urge other people interested to think about taking advantage of this. There's a lot of money involved, and it's a very interesting project because it involves using these lines derived from patients. So these are real patients with real cells. You make lines that you can differentiate into neurons and then ask, what are the defects in these neurons? Why did these people get Parkinson? You can use the different genetic environments, contexts, and this is very exciting work that shows how you differentiate, and then you get the cells. But I can't show this, and the other slides I had would not appear on the screen anyway. But we have used a lot the triplicate mutant in which you have overexpression. And the interesting finding so far is that when these neurons that have more synuclein start to differentiate, these are from patients now, they die. So the fact that you make the cell adopt the phenotype of a neuron is enough to lead to some catastrophic event or a series of events that kill the cell. And this is very exciting. We're trying to track it down with all sorts of means as shown on this slide. So I end with something that is really related to what we heard about yesterday from Luis Barreto, that cell-cell interactions are important. We think glia must be involved with this, and this isn't a glial cell and neuron, but these are two cells that are transferring Q dots with EGF on them, but we think that probably these contacts between cells are going to be very exciting in the study of these cells. So I hope that we can set this system up working here in Argentina so that the stem cell work can proceed. I have only the wish that we could facilitate, and we heard a lot about the expansion of science here in Argentina last night, but what we need to do, and I'm speaking now as an Argentine scientist, not as a German, so I can criticize a little bit, is to facilitate the importation of materials and equipment to make it possible to do this work. And not only that, once they get here, that they're delivered so that the cells don't die in the aduana or whatever. And besides that, it's possible in the universities to install air conditioners and trivial things like that so you can work with stem cells properly. So this I think is a new impetus, and one should add this to the list of logros that we heard yesterday from the President Kirchner, that to do science in Argentina can be more easy than it is now, so that we take full potential of this tremendous intellectual capacity that is represented by people in this audience. Thank you.